In lectures 1 and 2, I covered the 5 bases, and we looked at RNA and DNA, and we looked at de novo synthesis of the 5 bases, and we looked at the salvage of the bases to produce nucleotides. So video 1, which is this video, will cover the synthesis of pyrimidine nucleotides. These are the disorders. In this particular presentation, I'll be covering Orotic Aciduria, but there are 3 others, as you can see. Now, the 5 nucleotides are used to make RNA and DNA, and the bases are categorized as pyrimidine or purine bases according to their structure. So pyrimidine, the pyrimidine bases are made up of uridine, thymine, and cytosine, and the purine bases are made up of adenine and guanine. And you can say that uridine is present in RNA, and thymine is present in DNA, and the other 3 bases are present in both RNA and DNA. Right, this is a slide is to remind you about the naming of nucleotides. And it's important you understand how they're named, otherwise you're going to get confused because there's so many different names. There's adenine, adenosine, adenosine monophosphate, and so on. So this slide shows the structure of the nucleotide adenosine monophosphate. So this is the whole molecule here, but it's made up of components, 3 components. There's a base here, which is adenine, and there's a ribose moiety here, and then we have phosphate. So the base on its own is referred to as adenine. When you add a ribose to it, which attaches the C1 of the ribose, it becomes a nucleoside, and the name lengthens to adenosine. And when you add a phosphate to carbon 5 of ribose, it becomes a nucleotide, and the name lengthens to adenosine monophosphate. If you add another phosphate here, it becomes ADP, and if you add another phosphate, it becomes ATP, which stands for adenosine triphosphate. In the deoxy AMP or ADP or ATP, this OH here is removed, it's missing, so you just have an H. For the synthesis of the pyrimidine nucleotides, first of all, UMP is formed, and you can see this is based on the pyrimidine structure here. And from UMP, we can synthesize TMP by addition of a CH3 group here. And from UMP, we can also form CMP by the replacement of this double bond O with an NH2 group here. For the synthesis of the purine nucleotides, first of all, IMP is synthesized, and the structure of IMP is based on purine, and IMP is converted to AMP by the replacement of this double bond O here with NH2, and GMP is formed from IMP by the addition of this NH2 group right here. So there are 2 types of pathways used to synthesize nucleotides. One is de novo synthesis from new, starting with simple precursors, and one is a salvage pathway, which uses free bases from the food we take in and digest and break down. We use those to synthesize nucleosides, and there's a saving in energy if we do that. And there are some tissues that are very dependent on the salvage pathway. Not all tissues, just some of them. For example, the immune system relies heavily on the salvage pathway. Okay, so this slide summarizes the de novo synthesis of the bases, and it's good to have a clear understanding of what goes on, because then it makes the rest of the work we're doing understandable before you delve too much into detail. So this is an overview. We have the precursors here, and this slide is an overview for both the pyrimidine and purine bases. And from the precursors, and you can see they're simple precursors, PRPP stands for phosphoribosyl pyrophosphate, which I'll talk a little bit more about later on. But mononucleotides are prepared first, are synthesized first, and the mononucleotides need to be converted to trinucleotides. So there's specific kinases that convert a mononucleotide to a dinucleotide, and there's NDPK, which is a nucleotide diphosphate kinase, converts the dinucleotide to the trinucleotide. Now, this step here, the specific kinases, so each mononucleotide, be it AMP, CMP, GMP, and so on, each mononucleotide has a specific kinase. But for this step here, there's one enzyme that does it, which has broad specificity. And we need to produce the deoxynucleotides, and that work is done by this enzyme ribonucleotide reductase. It catalyzes the conversion of dinucleotides to deoxydinucleotides, and it works on 4 of the 5 nucleotides. It works on all of them except for thymine dinucleotide. And once the dinucleotides are formed, NDPK converts them to trinucleotides. So this is the synthesis of PRPP, phosphoribosyl pyrophosphate. The enzyme that catalyzes this reaction is PRPP synthase. And notice ATP is formed as used in this reaction, and ATP supplies this pyrophosphate here, which has high energy, and it's the hydrolysis of this later on that drives the reaction forward. So this is ribose 5-phosphate ribose, and this is the 5-phosphate here, and PRPP synthase catalyzes this reaction. This reaction is inhibited by the end products, ADP and GDP, and these are end products of purine nucleotide synthesis. It's also activated by ribose. So if we split our work now to look at the two pathways, it's to separate the two pathways, we see that pyrimidine bases are synthesized from these precursors, and the purine bases are synthesized from these precursors. They're quite similar, really. You can see the difference here is a glycine, and the difference here is carbonate. And one thing to know that with this pathway, as you'll see, the ribose, this comes into play once the ring is formed, and in this pathway, everything is built on ribose. So we start off with this. So that's a difference between the two pathways. And this pathway produces 3 nucleotides, and this one produces 2 nucleotides. So this summarizes the synthesis of the pyrimidine base. It's an overview. There are 12 steps altogether. Steps 1 to 6. We produce UMP from simple precursors, and then there's a branch point after step 7, where steps 8 to 9 converts UDP to CTP, and steps 10 to 12 converts UDP to TMP. This is probably why UDP is the one that is converted into its deoxy form, because it's common for both of these. Okay, so step 1, this is right at the beginning of the pathway. We have the precursors here forming carbomoly phosphate. The enzyme that does this is carbomoly phosphate synthetase 2, which is inhibited by UTP in red, and it's activated by PRPP and ATP in blue. PRPP the logic behind this really is that UTP is an end product, and it's quite common in biochemistry where the end product of a pathway inhibits the first step and sometimes the second step. So when you have enough UTP, you don't want the pathway to be active, so it's inhibited by the end product. And it's activated by PRPP. That's one of the precursors. So when you have an abundance of precursor, it drives the reaction forward. ATP is actually a purine. So the activation of the synthesis of the pyrimidine bases by a purine base balances the two of them. When the purines are plentiful, you want to produce the pyrimidines to balance them, because in DNA you need to have fairly equal amounts of the bases that actually bind together in DNA. And this is step 2 of the pathway, and you only need really to know steps 1 and 2 for this part of the pathway. And this carbomoly phosphate is converted to carbomoly aspartate by the addition of aspartate here. So aspartate actually forms part of the ring, and this reaction is inhibited by CTP, which is an end product, and it's allosterically activated by ATP, and that follows a similar logic to what I explained before with step 1. So we skip now from step 2 all the way to step 7. This is step 7 here, where we formed UMP, and UMP kinase, remember these are specific kinases, will phosphorylate UMP to UDP, and then UDP is converted to UTP by nucleoside diphosphate kinase, NDPK, and UTP is converted to CTP by CTP synthetase, and here we have the addition of this NH2, we have this amination. Note, CTP inhibits this reaction, so when we have sufficient amounts of it, the reaction is slowed down, which allows the balancing of levels of UTP, TMP, and CTP. UDP can also form TMP, so it's reduced first by ribonucleotide reductase to form DUDP, and then DUDP is converted to DUMP, which is then methylated to form TMP, and the enzyme there is thymidylate synthase. Okay, this slide really looks into, in more detail, what happens when DUMP is converted to TMP. And the reason why we're looking at this in more detail is that a drug is used to target this enzyme and a drug is used to target that enzyme. And there's the relevance of folic acid in this cycle here. So first of all, what happens? Well, we have the methylation of UMP to form TNP, and the methyl group actually comes from serine. Serine has 3 carbons, and it's converted to glycine, which has 2 carbons. So the extra carbon, ends up here as methylene, as the methyl part of THF. THF stands for tetrahydrofolate. So you have methylene tetrahydrofolate, which donates the methyl group and is converted to dihydrofolate. And once this has taken place, we need to regenerate the methylene so that this reaction can continue. So we have a cycle here. So dihydrofolate is converted to tetrahydrofolate by dihydrofolate reductase, and this NADPH provides the energy to drive this reaction. And this tetrahydrofolate can now be methylated to form methylene tetrahydrofolate. So the drugs I was speaking about, fluorouracil inhibits this enzyme here, dihydrofolate, and methotrexate inhibits thymidylate synthase. And the significance of this really is that if you inhibit what goes on here, you stop the production of TMP, or you slow it down. If you slow down the production of TMP, you slow down or stop the synthesis of DNA, and if you do that, then you stop cell division. Because when cells divide, they need to make a copy of their DNA. So dividing cells are affected by this. So that would be cancer cells, that would be hair, the immune system, any cells that are dividing. Methotrexate is commonly used to terminate pregnancies in the early stages, and it's also one of the most effective medications for the treatment of rheumatoid arthritis and other inflammatory types of arthritis. But both of these drugs can be used to treat cancer, which stops cell division. Fluorouracil can be applied to the skin to treat precancerous growths and also cancerous growth. Okay, so this is a little case study here. A young male presents with these symptoms here: retarded growth, anemia, mental retardation, and so on. The key thing here is excessive excretion of orotic acid in urine. So how does this come about? Also, this young man displays megaloblastic anemia, which is not cured by folic acid. Normally, a deficiency of folic acid causes megaloblastic anemia, because without folic acid, you can't make TMP, because tetrahydrofolate is low. And the red blood cells, they don't mature properly, and that causes the megaloblastic anemia. With orotic aciduria, one or both of these enzymes are defective, and that stops this pathway here, and it leads to a buildup of orotate or orotic acid, which accounts for its presence in urine. The type 1 deficiency is deficiency of both activities, and type 2, which is rarer, is caused by deficiency of only orotic decarboxylase. So how would you treat this? Well, from our knowledge of nucleotide mechanism, we can treat it, and we can treat it by administering uridine, which will be converted to UMP, CTP, and TMP. And so we get the 3 nucleotides that are required for DNA synthesis or RNA synthesis, and also UTP feeds back to inhibit step 1 of the reaction. And that means we don't get a buildup of orotic acid, because this pathway here, which leads to the synthesis of orotic acid, does not occur. And that's it.